for coming. Before I start, I wanted to give a special thanks to my parents for driving all the way from San Jose to watch a 15-minute talk. My name is Colin Kim, and I'm going to be a third year majoring in biochemistry in the College of Creative Studies at UCSB. I'm currently working as a MARC scholar in Professor Irene Chen's laboratory in the Department of Chemistry and Biochemistry. And this summer, I worked under the supervision of a graduate student mentor, Baoqing Zhu, on a project called Detecting and Quantifying Bacteriophage N13 on Human Skin. So these are some of the beautiful pictures of bacteria that I may be working with near down in the future of my research project. Because of the evolutionary adaptive abilities of bacteria to their environment, antibiotic-resistant bacteria are outcompeting the development of antibiotics in modern medicine. We are currently facing many problems with antibiotic-resistant bacteria, such as the recent discovery of a superbacteria called carbapenem-resistant enterobacteriolase in the water aquarium. And as biomedical researchers, we are hoping to find an alternative to treating these bacterial infections. Bacteriophages are natural viruses that infect bacteria, and the fact that they can target and kill specific pathogenic bacteria holds a huge potential for us to research about its usage on using it as a therapeutical method. Bacteriophages are non-living viruses with a single-stranded or double-stranded DNA or RNA inside of a protein code that is specific to its host bacteria. So we can utilize that to target specific pathogenic bacteria and eliminate our specific bacteria that are in the bacterial infections. In order to first develop the phage therapy techniques, we felt like the first appropriate step in doing that is to understand the interaction between bacteriophages and their host bacteria. And in my project, we are specifically looking to characterize the microbial community of human skin wounds, such as diabetic foot ulcers and surgical wounds. And as of now, we do not know the precise composition of bacteriophages and their corresponding pathogenic bacteria in these wounds, but we do hypothesize that there are many different pathogenic bacteria and their corresponding bacteriophages that make up these wounds. And in order to first characterize the microbial community of these wounds, we have to effectively recover and detect bacteriophages by swabbing the human skin. As of now, we do not have a standardized protocol in which we can do that. So this summer, I worked with a model organism called Bacteriophage M13, which is an E. coli phage that has been extensively studied. We know about its purification method, and we know about its genome. So we will be using this to develop a skin swab protocol in which we can effectively recover and quantify bacteriophages by swabbing a human skin. So the following methods that I'll be going over have been approved by the UCSB Human Subjects Committee. And first, we pipette a known amount of bacteriophage onto a volunteer's skin. And we will be using a cotton swab to swab off the bacteriophages from the volunteer's skin and use a sterilized tweezer to tweeze off the cotton from the cotton applicator. And the cotton will be resuspended in PBS buffer and vortex to resuspend any bacteriophages and bacteria that were swabbed off from the human skin. And we will centrifuge down the tubes to spin down any bacteria or contaminating debris and extract out the phages from the supernatant and transfer it to a different tube. And we will add DNAs to further purify the bacteriophages, removing any contaminating sequences. And we will have our isolated bacteriophage M13. Once we have our isolated bacteriophage M13, we will use the Purelink viral DNA extraction mini kit to lyse open the protein coat of the phages and extract out the DNA and purify it. Once we have our extracted phage DNA, we will use quantitative PCR, which is also known as real-time polymerase chain reaction, which works similar to a normal polymerase chain reaction that you guys might know. The targeted DNA sequence is replicated each cycle, But for qPCR, we're using cyber green, which interacts with DNA particles inside the solution. So the cyber green fluoresces and binds to the DNA each cycle. So we can utilize this to analyze our data and see how much phage we can quantify by swabbing the human skin. So far, as of now, we have collected many different rounds of qPCR data of our human skin samples. And the samples were generated by a 10-fold serial dilution of M13KE, which is a vector phage derived from M13 with similar DNA sequences. And for our control samples, we directly extracted the DNA from these serial dilutions. And for our skin swab samples, we dropped those serial dilutions onto a volunteer's skin, swabbed them, and extracted the DNA. So the samples that I'm about to present have been assayed in triplicates, and in this data we have calibrated these data points onto a standard curve with an R-squared value of 0.98. The standard curve was generated by using a known plasmid called M13MP18, which is also derived from the M13 family. And here I plotted the CQ value, which is the quantification cycle. And this represents the number of cycles at which the sample reaches the cycle threshold of fluorescence. And I plotted that against the log starting quantity of number of DNA copies in the sample. So here I plotted the control samples along the calibrated standard curve, and you could see a linear trend along the standard curve, which ensured that if we were to use qPCR to quantify stock bacteriophage M13, we would be able to effectively detect them and recover them. And this data allowed us to conclude that the qPCR baseline of detection was 2.36 times 10 to the 4 number of copies by using the no M13 control and using the linear equation from the standard curve to calculate that. Here I plotted the skin swab samples along the standard curve line, and here you can see a linear trend until the 5th diluted series where the CQ value was lower than the expected value. And this means that if there's lower number of CQ value, that means the qPCR machine is outputting a greater number of the total number of DNA copies. And from this data, we felt like we concluded that the limit of detection by using qPCR to detect bacteriophages from swabbing the human skin was set at 10 to the 6th number of DNA copies. And this is due to the contaminating sequences on the human skin that is inflating the qPCR signal and giving us more number of DNA copies than it actually is. So what does this really mean? So this means that when we are utilizing and using this protocol to quantify phages from human skin wounds, if there are more than 10 to the 6th number of copies of bacteriophages on human skin, then we could use qPCR to quantify it. If there is less than 10 to the 6th, then we cannot use qPCR to quantify it. So if there is less than 10 to the 6th number of bacteriophage DNA copies on our human skin wounds, then how do we quantify it? So that's where our feature work comes in. So when we have our extracted phage DNA, we will mix it with a 5X control plasmid, which has been known to have no overlapping sequences with M13KE DNA. And that will give us the ratio to compare it to at the end when we get our quantification data. So we will cut these into fragments, add the adapters to do a sequencing library preparation where we will do Illumina next generation sequencing to sequence all these DNA particles, use the Galaxy genome assembly and mapping to map these samples to M13 and 5X genomes, and we will analyze our data and see how much phage M13 we can recover from doing the metagenomic analysis. And in the future, we will be using this protocol for metagenomics to collect our samples and apply the phage detection protocol. And we will ultimately analyze the metagenomics and that data in the microbiome and see what kind of bacteriophages and their corresponding pathogenic bacteria are in the human skin wounds. So I would like to thank my funding agencies, NIH, CSEP, CNSI, and ICB for giving me the opportunity to spend this summer and do my research in the Chen Lab. And I would like to also acknowledge the MARC USAR coordinators for guiding me through this process. And last but not least, I would like to thank my graduate student mentor, Baoqing Zhu, for helping me and guiding me through the summer program. So, thank you. Any questions? Yeah.
Okay, so you know now that you have one method that's only good to about 1 million phages.
Right.
Do you have any expectation that, do you have any idea what a logical number might be in a wound as far as number of copies?
Yeah, that's a really good question. She asked how much do we really expect from, how much of the phage concentration do we expect from swabbing the human skin, or how much there is. We actually don't know the composition of bacteriophages and the bacteria and the concentration of them that make up those wounds. So that's why we're developing this technique to recover them and detect bacteriophages. We are hypothesizing that there are more than 10 to the 6th number of phages on those human skin wounds because they make up of a lot of pathogenic bacteria that make up the biofilm, and if there are many pathogenic bacteria in the communities of bacteria, then we assume that there are many different bacteriophages that make up those wounds as well. And we are doing further research and getting a more accurate representation of the limit of detection by doing metagenomic analysis, and we're hoping to push the limit of detection down so we have a more space to work with. Yeah, so that's actually very interesting because we actually don't know that much about phages in these human skin wounds. So we felt like this was the first step in generating a protocol to effectively swab and recover phages from. So,
can you go back to your figure with the calibration curve?
Yeah. So
going,
this one or the back.
Just keep going back. Here. So
yeah.
How did you determine that the baseline was 10 to the 4 when the control is actually 10 to the
yeah. So this is a no M13 control, and this plot, I should have mentioned that. This plot, I plotted the CQ value that the qPCR machine outputted with the expected amount of DNA we were supposed to recover. So expected amount is obviously 0 because we had no phage DNA, but we used this CQ value, and from the standard curve, they gave a linear equation. So I input the CQ value into that linear equation.
So 10 to the 4th was determined based on the linear regression?
Yeah.
Not from the observed data set?
So no, so I inputted the CQ value into the standard curve, and that gave 10 to the 4.
What was
0.36 times 10 to the 4.
Okay.
Yeah.
What was the error on that?
So I would have to check on that, like this error bar over here. I think I have the data towards the end.
It seems like it's pretty significant, right?
Right.
Close to an order of magnitude, but I mean, who knows? Oops.
Yeah.
Oh, there you are.
Okay.
Well, it's all good. Any other questions?
Yeah.
Just so to follow up on exactly what Stephanie was thinking. So qPCR is good, fast maybe.
Right.
NGS is awesome, not as fast. So do you see at some point that even if you find that your colony population is much larger than 10 to the 6, you might still use NGS just because it's so much more accurate?
Okay. Actually, for the skin swab, human skin wounds, the clinical samples, we will only be using next generation sequencing because we ultimately want to get the DNA sequences and characterize bacteriophages and bacteria from those wounds. We wanted to use the qPCR because it was a rapid method to detect